All right, I think I'll get started because we have other speakers after me. To begin, welcome. Thank you for joining. My name is Mark Keel. I'm the Chief Science Officer and founder of Genomic. And typically, I talk about clinical genetic diagnostic practice in the present tense. But today, we're going to talk about how Genomic is leveraging artificial intelligence and real-world data to make discoveries that impact the future of clinical diagnostics. So I'll be introducing the topic very briefly, but then I'll also be introducing my colleague, Jonathan Eads. Jonathan has 20 plus years of experience in AI as a practitioner, making discoveries in genetics and medicine and genetic medicine. He is our VP of engineering at Genomic, where we simplify complex genetic data to make actionable insights. We do that for clinical diagnostics, as many of you well know, but also for drug development. We offer data, software, and services to realize that value. We have a unique combinatorial approach, and Jonathan will be touching on some of these aspects. The first on the left is sophisticated computational techniques that allow us to find and refine the valuable insights from the scientific and clinical literature. And the second, often in a combination, 1-2 punch, is we have a large team of dedicated genetics experts who inspect and perfect that data to make sure the information is not only accurate, but meaningful. Today, we're going to be talking about real-world data, real-world evidence, and real-world insights. So everybody is undoubtedly familiar how clinical trials can influence drug development and influence clinical practice, but they're often missing the real lived patient experience that comprises real-world data. And by real-world data, I mean the individual data points that characterize their diagnostic and therapeutic journey, that when you assemble those together, form real-world evidence in the form of data sets to help make insight out of those patterns in the data. And so what Jonathan will be talking about is a valuable resource for that real-world data, which is to say the clinical literature. I mentioned the patients. The different forms of patients, as they're mentioned in the literature, come in the form of case reports. So the individual patient and their journey as it's cataloged in those publications, as well as in the pedigrees, the families, the probands and their siblings, but also the cohorts, the meta-studies, and the trials that are summarized in those publications to capture the demographic and phenotypic data, the clinical laboratory values, the treatments, and the outcomes. So this collectively is a great compendium of information and a great resource for making some of these discoveries. If you're familiar with Genomic at all, you're undoubtedly aware of Mastermind, which is a resource of curated genetic variants as well as indexed information focused on rare genetic disease across the whole exome. We also have a great valuable data set called the Cancer Knowledgebase, which is a curated compendium of those genetic variants, the treatments, and the trials associated with those variants for somatic oncology. So that's at the genetic level. What we're going to talk about today is widening that aperture to include that real-world data, the clinical information, the patient journeys, the treatments, and their outcomes to inform the discoveries that Jonathan is going to be talking about. So before I pass it over to Jonathan, the architecture of his talk is going to be to go through that technology and to reify that by way of example. So as he's going through that, I'd like you to focus your attention on three things. First is the value that lives within real-world data and the fact that Genomic is able to capture that value. Second, that we're able to assemble that into those real-world evidence data sets that I talked about. And third, that we're able to make valuable conclusions in the form of real-world insights. So Jonathan is going to go through that by way of example for colorectal carcinoma, but by extension, what he's talking about in that one example is applicable across every disease, including and especially rare disease, and is imminently scalable, which is part of the promise of artificial intelligence techniques. So with that introduction, I welcome my colleague, Jonathan Eads, who will be leading the conversation.
All right, thank you, Mark. So how do we capture real-world data from the clinical literature? The first step is to figure out what we're looking for, and what we're looking for are named entities that are relevant to clinical discovery. Named entities like gene symbols, genetic variants, diseases, phenotypes, patients, drugs, all of those would be the entities we want to be able to extract from the literature in an automated fashion. The process of doing that is natural language processing. We add another element to it. We don't just want the entities, we need the relationships between the entities. So if we were to identify a disease and a patient, that's interesting, but it's a lot more interesting if we can say which disease occurs in which patient, which drug treats which patient. So the relationships are critical for really extracting the value. There's two ways to do this. There's rule-based NLP and there's modern AI. Modern AI performs really well. This is something it excels at. Large language models can capture contextual relationships that traditional NLP cannot. They can also perform tasks. So, there we go. So here's an example of the kind of tasks an LLM can perform on the fly. The example on the left in the gray box is a copy number variant in natural language that was extracted from a full text article. So 12.9 megabase deletion at a particular chromosomal position. The data on the right of that is the output directly from our AI solution. And the first example there is a translation of that natural language description of a CNV into formal ISCN nomenclature. And that's a task an LLM can do. The other point of interest there are the assignment of genomic coordinates. So those genomic coordinates did not occur right around the reference to that CNV. They occurred somewhere at a different location within the article, a large word distance away. That's another capability of LLMs. They can make those associations correctly. There's a little table there that demonstrates our scores. Our accuracy scores for CNVs in particular are all in the 90% range. The last point of interest here is contextual awareness results in more accurate associations. The contextual awareness is critical. If we're talking about something like a gene symbol, that, the context in which it's mentioned really defines its meaning. Gene symbols could be the name of a plasmid or the name of a manufacturer. It doesn't necessarily have to be a gene, so that context is critical. So to accomplish this, we've built a comprehensive prompt and relationship schema. There's a little snippet of it. That's all the boxes and the arrows there. We can't fit it on one slide, but we've started our process going after 14 entities that we think are clinically relevant. Everything from genes, proteins, variants, diseases, individual patients, all the way out to epigenetics. Every box in that image has a prompt associated with it. And any one of those entities can have any number of nested properties that our AI solution looks for. I've highlighted individual patients. They could have a property of an outcome. An outcome could have a property of an outcome summary. Outcome summaries could be positive, negative, partial, complete, or progressive. So we can get really granular with patient outcome inference, and we can do the same type of thing for any of the other entities. We're also capturing 30 entity-to-entity relationships. So things like clinical trials test drugs, diseases occur in patients. Those relationship predicates we're using a standardized ontology to map to. I guess the main point here is we can find all of these entities and relationships in a single scan of an article, and we can scan the entirety of the clinical literature. So here's one example of normalizing a natural language description of a disease. The AI identified ulcerative colitis as a disease mentioned in a full text article. Everything in purple there are AI inferred nomenclature and context assignments. So the first assignment's a really critical one. We assigned an ID from a disease ontology called Mondo So disease mentions can come in a variety of different flavors. There could be different capitalization, different ways of saying the same thing. Normalizing to a single identifier really improves the value of that information. The AI was also able to assign an ICD-10 billing code and to give very specific context around the mention of ulcerative colitis. The second part of that context there, so targeted by the protective effects of colon ester. I'm not going to try to say the rest of that, but that's a very specific context of that mention, meaning we could query by that. The other piece of information inferred was the mode of inheritance. This is just one example of billions of entities from real-world data lit. We're doing this with all of the entities and all of the properties. We're mapping to things like the human phenotype ontology, HGVS and ISCN. We're converting into those formal nomenclatures. So what can we do with the prompt and relationship schema? Our whole intention here is to convert real-world data to real-world evidence. And when we extract these entities and relationships, we can place them in a graph database, and we can query that graph database to derive real insight. The other thing we can do with this is the entity and relationship schema, it's not limited to just real-world data lit. We can run it through any kind of text-based real-world data. Things like electronic health records, clinical trials, patient registry data, and we can combine that with what we derive from real-world data lit as well. So there are some challenges using LLMs to do this type of work, but probably one of the biggest ones that's in the media are hallucinations. We found we can avoid that for the most part by really sticking to text extraction. All we're asking the LLM to do is to extract specific parts of text or summarize natural language in different forms, maybe a controlled enumeration of strings or another shorter, more concise natural language summary. Probably a very important challenge is insufficient accuracy. The accuracy here is critical. And one of the ways we address that is we work with our curators and domain experts to assemble truth sets. So collections of papers that focus on where we've curated the entities and relationships we want to target, and then we can test how the AI does on that truth set. And when it's off, we can go back, adjust prompts, and this process is iterative. And we found if we stay at it long enough, we start driving accuracy into the 90 percentile. The other technique there, especially for getting normalized entities, is a AI technique called retrieval augmented generation. We make a lot of use of that, and it works well. So Genominon's historical focus has been on genes and variants. We are broadening that aperture to include a whole host of clinically relevant entities, things like patients and the drugs that those patients were treated with, diseases and therapies, clinical trials. Why are we doing this? 85% of Mastermind users have indicated that they would integrate this type of search into their diagnostic workflows. 73% of Mastermind users have indicated that they would be using this type of capability multiple times per week. So what are we really going for? We're going for dramatically improving the potential for high impact real world insights. And of course, we're going to be able to do that better when we have more clinically relevant entities. All right, we're going to take a break from the boxes and arrows, and we're going to look at some real data, a real case study on colon cancer, and see how this performs. So colon cancer patients treated with immunotherapies exhibit a wide range of responses, spanning from no effect to complete remission. Our question is, can we identify meaningful correlations in patient outcomes to immunotherapy treatments in colon cancer patients utilizing G3 and our prompt schema? We started by identifying full text articles that had genes, variants, and colon cancer mentioned together in our 10 million full text article corpus. That got us down to 50,000 full text articles. We further filtered by publications later than 1990. That's about when immunotherapies came on the scene. And then we have an additional AI model that can assign labels like clinically relevant, functionally relevant, or review to the articles. That got us down to about 14,000 full text articles. We scanned those articles with this prompt and relationship schema. That yielded 530,000 clinically relevant entities and 610,000 relationships. Out of those 530,000 entities, 5,593 were individual patients with colon cancer. Of those, 459 were treated with immunotherapies. And that forms our patient cohort that the rest of the analyses shown are going to be focused on. All right, so the bar chart on the right there shows the distribution of the entities that we identified. The most common ones, no surprise here, were genes and proteins. Those bars are not unique genes and protein mentions. They're all the genes and protein mentions in the publication, so we're expecting those to be really high. But we see pretty good coverage of our full set of entities. We see phenotypes and diseases and therapies. The two blue bars there, the smaller one, are the individual patients we found. And right next to it are patient cohorts. So patient cohorts referred to in the literature. The individual patients are where we're going to focus because we're able to infer outcomes from treatments at the individual patient level. The visualization on the left is the result of executing distributed stochastic neighbor embedding, so TSNE. It's a dimensional reduction technique. If you're not familiar with it, we put in a bunch of data, we collapse the data dimensions down into 2 dimensions, and in that visualization, each point represents an actual patient with colon cancer that was treated with an immunotherapy that was discovered in the literature. And the points that are closer together are patients that have more similarities. So the color scheme applied here was an outcome of disease progression. And you see a cluster on the right, that's all in red, that patients that exhibited no progression. You see a cluster on the left in yellow that exhibited disease progression. And then there's a blue cluster unspecified. So that's where the AI was unable to assign some kind of patient outcome based on the natural language in the article. And then progression not reported is in green in the bottom, and that means that the authors reported an outcome, but it's not disease progression. The last point of interest is the treatment failure. There were a few examples of that, but we don't have quite enough data to get a full cluster. The data that went into making those clusters was just patient outcomes, and there's a lot of them, and the drugs they were treated with. And remember, the patient outcomes, the AI is inferring those from natural language descriptions around the patients. The outcomes are a set of structured enumerations that are controlled. So we're forcing the AI to take natural language and give it a label of, disease progression relevant type things. So we're going to continue on studying these 2 clusters, but we're going to look at some specific facets of non-responders. So same set of clusters, but the panel on the right are the non-responders. Those are patients that did not respond to the immunotherapy treatments. They're colored in yellow. The panel on the left is the full set of clusters that were responders. The orange box there is where the non-responders would sit if we superimposed them. So what do we see here? We see a really well-delineated linear cluster with a high degree of similarity. And that was pretty surprising and also pretty exciting. I was expecting that to be a lot messier. That is a very well-delineated cluster. And remember, the data that we fed into that dimensional reduction includes the drugs and the patient outcomes. We're going to throw some more data in that dimensional reduction, but just those 2 buckets of data yield that kind of delineation. So same game, we've got the non-responders on the right facet, the responders on the left facet, but we're going to overlay treatment responses. So we see 3 treatment responses that map really well to the non-responders. No response to the treatment, shown in red, negative response in gray, and progressive disease. that's pretty much what you'd expect to see there. Mapped over the responders, we see complete response in green on the right side, and right next to it are the positive responses. And then we see mixed response, partial response, and stable disease dispersed. They don't really form strong clusters. This was really encouraging. That's pretty much what we were hoping for. Same game again, but now we're going to look at survival. So mapping an outcome of survival onto the non-responders, we see a no survival cluster mapping pretty well. There's some bleed over into the responders, which is what you'd expect with this kind of data. On the responder side, we see a cluster with overall survival, right in the same location that we were looking at positive treatment responses on the last slide. And that's what we were hoping for. So this was really exciting to us. Remember, these are AI-inferred patient outcomes from natural language descriptions. And we're seeing really good cohesion, really good delineation of clusters, and really solid evidence that we can use AI to extract a patient cohort automatically from the literature for any disease. In this case, it's colon cancer, but this will work with any disease. So we're going to shift gears a little bit. Instead of looking at responders and non-responders, we're going to facet by driver mutations. So in cancer, there are key driver mutations associated with oncogenes that are really responsible for ultimately the proliferation of those cancer cells. Each patient data point here is colored by an oncogene that is known to have a driver mutation relevant to colon cancer. The light blue patient colors are ones where the authors did not mention any kind of oncogene or driver mutation. When we look at the panel in the middle, so driver mutations true, not much to write home about there. We don't see any clusters, we see a lot of dispersion there. When we look at driver mutation false, we see a distinct cluster of a particular gene, KRAS. That's pretty interesting. It's also positioned, if you look at that first panel, we've got a orange box around our non-responders to keep the visual orientation there. It's oriented to the bottom of that box. So wanted to drill down and see what's going on there. And now the facets, we're going to go back to disease progression. And we're going to see where that KRAS cluster sits. Well, the entire cluster sits in the disease progression facet. All of the KRAS examples where the driver mutation was false also are patients that exhibited disease progression. Last bit of information here. We wanted to throw in some genetic information, and specifically the counts of single nucleotide variations and structural variations associated with the oncogenes. And in this case, all of the KRAS examples had one single structural variation. That one single structural variation was annotated as RAS amplification, specifically RAS amplification without RAS mutations, which is a common occurrence in metastatic colorectal cancer. So what's going on here? Well, the authors of this paper were studying RAS amplification as a negative predictor of benefit for anti-EGFR MABs. They were studying specifically 2 immunotherapies. I'm not going to try to read the names of those, but this explains why we see a KRAS cluster. All of the patients where KRAS was observed were also treated with EGFR MABs, and they all exhibited disease progression, which explains why they clustered in the disease progression facet. The authors reported RAS amplification without RAS mutations, which is why we observe KRAS with driver mutation false. The authors suggest that RAS amplification without RAS mutations may have poor outcomes on EGFR MABs. And that's kind of expected. So the mutation that is there is really causing KRAS to just overexpress. There's no mutation that would present as an antigen that the immunotherapies can interact with. And that explains why we're seeing it in the disease progression facet. So these are some powerful observations that you can't get without looking across thousands of publications at the same time, extracting the entities and extracting the relationships utilizing this kind of AI solution. We're just starting this analysis. We haven't really explained why we have such a beautifully delineated cluster of non-responders. And the fact that we got that cluster by just putting in the diseases and the patient outcomes, I think there's an answer there, and we're pretty excited to dig even deeper in this data set. There's a lot more to look at. We've also got a lot of phenotypes we could align with the genes and look for patterns there. This is an exciting new way to approach the literature. So it works, and it works well. We were able to utilize AI to identify a patient cohort for a specific disease. We identified 459 individual patients. We were able to, with a high degree of accuracy, map patient outcomes for drug treatments to individual patients using an AI solution. The important point here is we can do this for any disease. We never asked the AI any specific questions about colon cancer. We asked it to find every disease, and we pre-filtered for colon cancer because that's what we wanted to ask about. We could do this for any drug. It's not specific to immunotherapies. Any kind of drug class we can go after. And it's also not specific to the literature. So any kind of text-based information like electronic health records, clinical trials, patient registry data, we can go after with this prompt and relationship schema. So next steps, we want to release this within the year. We're going to scan our full article corpus, and we're going to put that into a giant graph database. Our intent here is to derive valuable real-world insights that you otherwise couldn't get to. We're going to release this in a product called Mastermind Discovery that'll provide a search interface for G3. We're also going to release a custom G3 scanning service that would allow you to provide your own query in addition to this set of entities and relationships we're going after. Some diseases have very specific entities and relationships. We're not going to be able to get them all generically, and we want to be able to go at very precise ones that folks are interested in. And the last bit there, you can probably guess, we want to expand this beyond the literature and go after electronic health records, clinical trials, patient registry data. Combining that with what we pull out of real-world data lit, I think is going to enhance the ability to have impactful observations that make a real difference. And that's it. A quick thank you to the team that worked on this data. Brittany Jones, Kalechi Fletcher, and Angel Moreau. We all worked really hard for the last couple of weeks putting this together, and we're super excited to see where it goes. And I think I have a time for a couple of questions. If no one has any questions, I have a ton of questions. Yeah. So the question was, would your model work for real-world clinical notes? And the answer is yes. Any kind of textual input this model will work on. The only things it won't work on, well, we can do this in other ways, but like image data or binary files or something like that. But we can, there's ways to work around that. Any other takers? Oh.
Hi, great presentation. I was wondering, since I guess like more data you put in the system, better results you get. What do you do with rare diseases when you don't have that many publications, like in cancer, for example?
We would be limited to the representation of that data in what we're scanning. So If it's not well represented in the literature, it's unclear whether we'd be able to find it or not. But we can get down to finding a single article. So it doesn't have to be well represented or overrepresented for this to work. But we're not going to be able to magically come up with a giant patient cohort out of thin air. It's going to be dependent on the input sources we scan.